Hello, everyone, and welcome to another episode of ALS Clinical Trials Unboxed, brought to you by ALS Canada. My name is David Taylor. I am the Chief Scientific Officer at ALS Canada. And today we have a special episode involving Coya Therapeutics. Actually, this is a really cool one because I had just been relaying to Daniel Barvin, who you'll be introduced to in just a second, that back during the pandemic, Coya had just sort of started out with clinical trials with something called Coya 101. And their CEO at the time was coming to Toronto for personal reasons. And I happened to just be able to manage to connect with him. And he was the very first person I had a socially distanced coffee with to talk about how can we get Coya to come to Canada to run your clinical trials. And of course, at the time, very small company running their trials out of Houston and Boston. And he had said, Canada's definitely on our radar for the future. We certainly can't do that right now, but we certainly know that you're here. And here we are with Coya 302 at several Canadian sites and here on Clinical Trials Unboxed a few years later. But so glad to have you. And with us today to speak about the clinical trial, ALSTARS clinical trial, is Senior Director of Clinical Development, Dr. Tyrell Simkins, and also the VP of Operations and Patient Advocacy, Daniel Barvin. So without further ado, I'm going to pass to them. We'll come back. And if you have any questions, please put them in the Q&A. You can use the chat, but it will be better if it goes through the Q&A. That way we can make sure we have a look and tick off anything that we do manage to hit. But otherwise, I'm going to pass to Tyrell, and thank you so much for being here, and we'll chat in a bit.
All
Pass it
right.
off.
Well, thanks, David. Welcome, everybody, and thank you for this opportunity for us here at Coya Therapeutics to address everybody and have a chance to give some information on what we're doing. So, here at Coya Therapeutics, we're focused on addressing neuroinflammation using combination therapeutics for neurodegenerative diseases, but very specifically today, we're going to be talking about our work in ALS. Very brief introduction by David, which we appreciate. Very briefly, though, and again, my name is Tyrell Simkins. I'm Senior Medical Director here of Clinical Development at Coya Therapeutics. I'm a neurologist and a neuroscientist. I'm very passionate about trying to find cures and therapeutics for ALS. And with me today is my counterpart, Daniel. I'll let him introduce himself.
Sure. Daniel Barvin, VP of Operations and Patient Advocacy. I've been with Coya since the very early days, and prior to that, have helped build and the legacy and a deep family history in being a C9 carrier. So I know intimately what this disease can do and how desperately we need to end it.
Well, thanks, Daniel. And he's an inspiration to all of us. And all of us here at Coya Therapeutics, we're a small company, but very dedicated to this cause. So again, we're very happy to be here. Some mandatory disclosures, which I'll put up there that we have to as a company working on developing a therapeutic. But we're going to get right into it. And so this is where I'm going to stop. And it's the slides that we've prepared today, they have a lot of information, some kind of technical information for those that, the ALS community tends to be pretty informed. But if that's not something that you want to dive into, you can really just listen to me. I'm going to be talking to this, but these are, there's information on the slide, extra information if you want to view it as we're going through. So want to start off by setting where we are and like our view of ALS and what we're doing. So we know that inflammation plays a critical role in degeneration. And so the left-hand panel, what that's trying to depict that in neurodegenerative diseases, and so we're talking about diseases like Parkinson's, Alzheimer's, frontotemporal dementia, and ALS, there are some very specific cells within the brain and the spinal cord that are dying. And when that happens, we call those neurodegenerative diseases. When that happens, there's inflammation. It's the body's natural reaction to injury or damage. It serves a purpose at a certain point to kind of help clean up things and trying to limit the damage. But what we've also found through years, decades of research, is that neuroinflammation from degeneration of these neurons can continue in this kind of feed forwards, like this cycle that can, at a certain point, stop being helpful and be actually damaging to the brain. And we found that this is a component that is involved in essentially all neurodegenerative diseases, but very specifically, we know in ALS that this neuroinflammation continues to drive some of the ongoing symptoms of the ongoing damage going on in the brain of the spinal cord, leading to the symptoms of ALS. And our bent or our approach is to try and address that to stop this ongoing neuroinflammation. And very specifically, there is an important cell called a regulatory T cell that plays a central role in that. What are T regulatory cells? So this is a long, decades of work by a very famous gentleman, a neurologist, neuroscientist named Stanley Appel, working on this specific cell type. So T regulatory cells are a very specific immune cell type, and their job or their role in the immune system is to balance inflammation. So there are times when we need inflammation, but we also are supposed to be able to stop it. And in ALS, we've actually found that T regulatory function is dysfunctional. They're not working like they should. There's less numbers and they don't work as well as they should. And so what we're doing here at Coya is to try and design therapeutics to boost or help out those T regulatory cells, to restore their function, to use the body's own systems to stop neural inflammation and restore normal function, and in this case, to improve or to stop the progression of ALS. With that, I'll introduce our lead candidate right now, Coya 302, which is a combination product of something called low-dose interleukin-2 and CTLA-4 IgG, or also known as abatacept. So again, technical slide, but I'm going to walk through this. So this is what Coya 302 is. So on the very left-hand panel, you'll see that there's a depiction of a dysfunctional T regulatory cell, which is happening in ALS. When you give the body this, it's a protein produced by the immune cells called IL-2, and you give it in low doses, it actually turns to make those dysfunctional T regulatory cells functional. It improves their function and it expands their number. A good thing, what we're talking about, like what's been happening, what we think is happening in ALS. What we've also found out through years of research and other clinical trials is that just stimulating T regulatory cells is not quite enough. There's a lot of inflammation. There's a lot of things going on in the body. And that's the middle panel. And so there's a very important cell type in the immune system called the macrophage, and they can have different functions. But one of the things that they do is that they can drive inflammation. So they can continue this process of inflammation going on in the brain and in the body. And there is a drug there's already out there that we're using called CTLA-4 IgG, or abatacept. And the purpose of that is actually to take those pro-inflammatory, those inflammation-driving macrophages, and then turn them into anti-inflammatory macrophages, a different type of macrophage that reduces inflammation. And that can happen throughout the body, but also they go in and have some effect in the brain themselves. So we're, to use that, I'm a child of the 1980s, you do a Voltron, take these individual pieces and put them together and then work together to have a desired effect to expand and improve the function of T regulatory cells and stop neural inflammation, and in combination to stop the ongoing inflammatory destruction or injury that's going on in ALS, and to stop the disease. With that concept and the research that came out of this, one of the things that I wanted to present to everybody is a early proof of concept study that led to what we're doing here at Coya. It was done again by Dr. Stanley Appel and his colleague Ali Faradar down at Houston Methodist. And in which they took this combination and they tested it in 4 patients with ALS. So this is kind of the scheme of how they did that trial. So they found 4 patients insured, and they had diagnosis of ALS, and they measured how quickly they're progressing. And so one thing that we use in our clinical trials, if you're not familiar, is a score called the ALS FRSR. It's the ALS Functional Rating Score, and then it was revised for some improvements. And it is a questionnaire that's administered by a physician or a clinical trialist to measure how ALS is affecting multiple aspects of your life: speech, swallowing, movement, and breathing. And so we're able to measure, and we have a lot of data on how patients with ALS have changes on that. So we ensured that when we got these 4 patients, we knew how quickly before coming into this trial that they were losing points on the ALS FRSR, progressing. And then they were given this combination of low-dose IL-2 and abatacept or CTLA-4 IgG for 48 weeks. During that time and at the end, we measured safety, we measured T regulatory function and number, and very importantly, how were they changing or what was going on with their ALS FRSR? How were their symptoms changing over that 48-week period? The takeaway from these 4 patients who were given this combination as a proof of concept are essentially 3. One, the combination that's being the 302 combination enhanced T regulatory function. In the top graph on the right-hand side there, on the y-axis or the up and down axis is a measure of how suppressive these T regulatory cells are, how much they can reduce inflammation or stop the immune system from attacking itself. And you can see as you go into the bars across the right, you can see that at baseline, there was some level of it, but when you started giving the Coya 302 combination, there was an increase in their ability to suppress the immune system, which is what we want, to turn off neuroinflammation. Also, the second takeaway is that not only did it increase their suppressive function, but it expanded or increased the number of T regulatory cells. It's not on there, but I'll show you that we had good measures of that. But maybe what's most important, particularly from the patient perspective, is like we're able to show that it's doing what we think it's going to do in terms of the T regulatory cells. But if you look at that bottom graph, that is a measure of those 4 patients and their ALS FRSR score. When they started the trial at baseline, their ALS FRSR was on average about 33 out of 48. And as I told you, we knew the rate at which they were progressing before they started getting the Coya 302 combination. They were losing about 1 point per month. But what you can see on that graph there is that out over even 56 weeks, they were treated for 48 weeks and then followed for another little bit, that between when they started and when they finished Coya 302, they had essentially no change in their ALS FRSR, showing that in these 4 patients, that it suggested that we were able to halt the progression of ALS. Very, very encouraging. And with that data, we thought we moved on to develop the ALSTARS trial, a larger trial to test the effects of Coya 302 in a larger population. So this is the trial that's ongoing right now and is enrolling in Canada. So things to think about. When we design a clinical trial, we have to do inclusion and exclusion criteria. It's not to exclude people, but it's to exclude people unnecessarily. But we really have to design the trial to show how the drug or the therapeutic we're doing can have an effect. So the inclusion criteria for this trial is patients have to have ALS, diagnosed ALS. Their symptoms have to have started within just over 2 years, 28 months. They have to have a baseline ALS FRSR score of 35, and your clinician, the physicians that you talk with at the clinical trial, they'll help you work through that. And the reason is that we have to be able to measure how things are changing over the time. When you enter the trial, we have to know how quickly the ALS is progressing so that we can have some idea of what to expect over time. And then we look at their respiratory function again, being able to measure how it preserves things over time. So once patients are determined to be included or eligible into the trial, they're enrolled and they get randomized for the first 6 months into one of 3 categories. Part of science and understanding if drugs are going to be effective is that there has to be a placebo group. But placebo is only 1 out of 3 groups. So there's a placebo group, but then there's 2 different regimens for the Coya 302. It is getting Coya 302 once a month, or it's getting Coya 302 twice a month. And we're testing 2 different doses to see how patients do and if one is better than the other. Importantly, after that 24-week period, we're going to take what happened at baseline and what happened, and at the week 24 time point, measure what happens in the ALS FRSR over that time. What's the progression of the rate? But then after that, for another 6 months, all patients in the trial are going to be on some form of 302. The placebo patients will be put into one of the 2 categories of either once or twice monthly dosing, and everybody that was already on 302 is going to continue to be on it for another 6 months. This is so that everybody has an opportunity to be on Coya 302, get the benefit as we see it, as well as be able to measure what's going on. And we're looking at safety, efficacy, and then how much decrease or how much slowing of ALS we can get with Coya 302. That's the design of the trial. I'm going to turn it over to Daniel to talk about some more specific things about the trial and how it's designed and some key features that we think are beneficial to the ALS community.
Thank you, Tyrell. So, with the ALSTARS trial, we're thrilled that the route of administration will be a subcutaneous injection. People might be knowledgeable of how diabetics have been treating themselves with insulin or of the more recent GLP-1 and how those have been administered. Subcutaneous injection is a small syringe, about a half of an inch or smaller, put into the subcutaneous fat. We will be having patients administer these shots in this protocol, which I'll explain in a second, in the following areas in the abdomen, around the navel, the back of the upper arm, or the lateral thigh. These are places where the pain will be the least. There's the most fat that can absorb the shot and be the most comfortable injection site for patients. That's why we chose those. Patients will be mixed through a dosing regimen of either one injection per month or two per month. And you will follow this route of administration. On day one, we'll have patients undergoing a abatacept shot in combination with a low-dose IL-2 shot. The abatacept will only be administered 1 day per week, while the IL-2 will be administered 5 days a week. And depending on which cohort you're in, you'll be doing that once or twice a month. Tyrell, next slide, please. At Coya, we have been really focused on how we can make this trial as easy as possible for patients, as we ourselves, some of us are. And to that end, we've been focused on taking some of these matters into account. Firstly, we're having a 2-to-1 randomization to treatment. So we'll have more patients on therapy than on placebo, which we are thrilled to bring to patients. And like Tyrell said, we'll be having our active blinded extension. So all patients will be on treatment after the 6-month period. Again, our route of administration will be subcutaneous, not an IV or intrathecal. Having had a few spinal taps myself, they're not the most comfortable. And patients will be allowed to stay on other therapies. I do also want to mention that we are agnostic to disease onset. Whether your ALS is sporadic or comes from a known genetic cause, patients are eligible for this trial if they have a diagnosis of ALS, regardless of how it came, if they meet inclusion criteria. Again, our endpoints will be ALS-FRS, and secondarily, we'll be focused on neurofilament light, respiratory endpoints, and survival. Next slide, please, Tyrell. We are thrilled to partner with a few of the doctors in Canada, Dr. Gedge, Dr. Shoesmith, Dr. Zinman, and Dr. Pioro. And the best way for patients to determine their eligibility for this trial will be to reach out to sites directly. If patients navigate to trials.coyatherapeutics.com or if they go directly to clinicaltrials.gov, they can search for our NCT number, or you can just search Coya 302. You'll pull up our trial listing and you'll scroll down to find sites. Please contact the site nearest to you. The contact information will be at each of those websites for these Canadian sites. And the clinical trial coordinators will respond. If they don't, please reach out again and pester them. But they will respond and they will schedule all of the screening necessary to determine eligibility. We hope, and we know the power of the patient community and how your voices can share the information about the trial being open and enrolling to your friends, to the community. And we want this trial to enroll as quickly as possible, as I know that this burden of disease with no treatment it needs to end. So we want to be sure that this trial enrolls quickly so we can have our data read out and we can see where we go from here. So we appreciate all of your support and help in connecting with patients and connecting them to our trial. With that, we want to thank you all for joining and taking the time to hear about Coya and the ALSTARS trial, and we are open to questions. Thank you so much.
Fantastic. Thank you, gentlemen. That was a wonderful overview. Really appreciate it. First time and maybe the last time I'll hear Voltron being mentioned on one of our webinars. So, as a
Hopefully not
person,
the last time.
yeah, hopefully not. I mean, we could talk about Voltron for hours, right? But thank you for this. And we definitely have some questions in the chat. So I'm going to hit those up first because I really want to make sure those people are getting some of the answers that they want. And then I'm gonna, we'll get into a little bit of chatting about some of the other things that I definitely had in there. So first of all, someone was asking about whether or not there's any exclusion criteria about someone with bulbar onset ALS, and not being familiar myself with the whole set of the criteria, are people still being able to be enrolled?
Great question. Yes. So bulbar ALS is not an exclusion criteria. We have you can have bulbar ALS. You can have limb onset, bulbar, even respiratory onset. You just have to meet the other exclusion criteria, but that's not an exclusion.
Great. And another question was around how long a patient needs to be off trial before they might be able to be on this trial. Whether there's any, I would add to that, which people might wonder, are there any specific off-label treatments or other trials that they might have been on that could exclude them? And then maybe you could just allude to, since we're here, I know there's just some careful stuff around when they may have started as standard of care therapy around riluzole or edarvone.
Yeah, great questions. No, and questions we're prepared to answer. So I'll hit it in two aspects. So we'll talk about first, like if you were in another trial, what is the time period that you need to be done with that before entering into? So depending, there's a couple of different versions of like how that goes. The standard answer is that you have to have been off of another therapeutic, like in another trial, for at least 30 days or what we call 5 half-lives. If you've been given a drug or a medication, we measure how much is in your blood by a half-life, basically how long it takes for it to get out of your blood by half. 5 half-lives, whatever that means. So if it were, 2 days of a half-life, then you have to be off for at least 10 days. And so you can do that math, or 30 days, whichever is the longest time period. And the reason for that is not to be punitive, but is to make sure that we get a clean or a good assessment of what is happening with Coya 302. A couple instances, things that have come up that is a little more challenging is that there are investigational and off-label uses of stem cells for different types of injections. That one is trickier just because you can't measure the stem cells themselves, but we do know that stem cells themselves have some effect on the immune system. And so that requires a much longer time period, probably more like 6 to 9 months that you have to be off it so that it doesn't interfere with what we're actually doing here. So that would be in terms of like other investigational trials. The only other exclusion that we have is if you're on more than 10 milligrams of biotin a day. You can be on biotin, but not more than 10 milligrams because there's some investigational things that are going on there and we can't control for that. To talk about the standard of care therapy. So for edaravone and for riluzole, you have to be on a stable dose, and have been receiving it for at least 30 days with no plans of changing it after that point. If you're on it, we want you to be, on it and stable at that point. With regards to tofersen, we require a 90-day period. So as long as you're on the therapy and stable for 30 days for riluzole and edaravone, or 90 days for tofersen, then you are eligible and can be enrolled in the trial. There's no restriction, for instance, for Nudexa. So.
And we do not have new Dex in Canada anyway. So,
Yes.
but I suppose, some people do compound it.
Yeah.
Actually, that actually hits off on another question that was up and I just got removed, but it was around tofersen. It's an interesting aspect that you considered people on tofersen, and out of just curiosity, for example, let's say you enrolled one person on tofersen, but they happen to be in the placebo arm.
Yeah.
Already a small trial overall with 40 people per arm, knowing that that's an effective therapy, that's a bold choice. And I was wondering, I'm sure you thought about that extensively, and it might be more just around, Daniel's, really strong intent to make these community-friendly trials, and maybe that's what the reason why you went with that in the end. But I was wondering if maybe you could expound on that a little.
Yeah. Two parts of that. So, first of all, part of that was to be patient-centric, and I don't know if you had anything to necessarily add about that. Daniel's getting over a cold, so first of all, I'll give Daniel a couple of seconds to talk about like our approach and patient centricity, which did play into this.
So, tofersen has been a real breakthrough for patients. What it has done for the SOD1 community has been revolutionary. However, it is only effective in a very small cohort of patients with a very particular type of SOD1. So it's not the right term, equitable for the patient community. And we want patients who are SOD1 carriers to be able to enter our trial. We don't want them to be excluded. So this was the main point of why we allowed tofersen. We see a large amount of patients being having an effect with tofersen, but a lot of patients don't respond. And so we want to be the solution for those who are not responding.
And in terms of how we handle that from a scientific standpoint, is that any patient who is under standard of care therapy, whether that's riluzole, edaravone, tofersen, we do take in whole and we do a statistical analysis on like all patients, but we do have sub analyses to control for those that are on standard of care therapy. So statistically and case by case, we can look at that and are well equipped to handle that.
Great. And thank you. And I want to actually dig into that in a moment, but I want to get back to some of the questions that are in the Q&A. There was a question from a gentleman around someone in New Brunswick taking this in Fredericton. You can speak to that if you'd want, but I think just to say that, companies have to choose certain trial sites for a lot of different reasons. The site in Fredericton has a phenomenal ALS leader. You never know where things go in the future, but right now, when it's available only in those sites, those are the only sites that you'd have to reach out to and go to. But if you wanna double down on that at all, either of you, please feel free to. Just I think it's on ourselves at ALS Canada to continue to also help people understand why certain sites get trials and certain sites don't.
Yeah, it's a great question. If it were up to me and unlimited resources, absolutely, I would be at every center in Canada or the U.S. or around the world. But we do based on criteria like how well equipped the site is, able to do the things that we need for the trial, and the staffing resources and the people that we work with understanding it. So that's understandable. And so that's part of the reason why we do that. One thing I will say, though, that if you contact these sites, we do offer some reimbursement for travel. And we, again, from the patient centricity and equitable distribution or equitable access, we're trying to overcome some of those barriers that way. So I would encourage you to reach out to the site and see what is able, and if the site in Montreal, for instance, or one of them is closer, you can talk with them about like feasibility.
Yeah, and I think and that's all really good points too. I think in a lot of these cases, it's hard even on a webinar for you to be able to determine when someone's asking about eligibility, because that's what a lot of people want to know.
Yeah.
On a case by case basis, you're not going to be able to know. So it's again just reiterating, if you're interested in participating in this research, please check out any one of the opportunities. Reach out to ALS Canada, reach out, I'm sure, to Coya as well. They can tell you the contact information for the sites. It's also on clinicaltrials.gov if you want to go there and reach out to the site and find out before you go there if this is something that you might be eligible for. They're going to be able to help you with your unique situation to be able to determine if you are eligible for the study.
And David, let me say quickly,
Sure.
Coya, ALS Canada, the only people that can actually determine your eligibility is the clinical site. That is the people to talk to determine eligibility. We can answer very broad questions, but Coya does not determine eligibility for the trial. It is only the actual sites that do so, and the best and quickest way to access screening will be through contacting the clinical sites listed on trials.coyatherapeutics.com or on clinicaltrials.gov.
Yeah, and thank you for clarifying that. Yeah, from the standpoint of reaching out to us, it's mostly just to get contact information for the sites.
Right.
And just so everyone's aware, and this isn't always obvious to everyone. This also keeps at arm's length from companies, right? In terms of, academics who are running the trials, right? A company has a vested interest in getting a result, and they're a business. And so when it's run in tandem with academics, the academics will, those sites can run that based on a protocol, and it's at arm's length from the company and how it's run, so that when the company gets the data back, they can analyze that knowing that it's as minimal bias as you'd want to insert in that. So you get a ground truth answer. So I appreciate you clarifying that
Yeah.
Daniel.
Sure.
So I guess can ask a couple of quick questions. There is a question around blood-brain barrier because it's a subcutaneous injection. I'm assuming yes, you've clarified it's going to get into a bioavailability to the CNS.
Yeah, so no, that's an amazing question, and this is where understanding the immune system in and of itself. So, low-dose IL-2 and the abatacept, the combination therapy, we actually don't believe gets into the blood across the blood-brain barrier. There's possibility, but it doesn't have to get across the blood-brain barrier because what we're really trying to do is we're trying to change the immune system, which is more majorly housed or is housed in the rest of the body. And then those T regulatory cells and those anti-inflammatory macrophages actually move into the brain and can have their effect. So that's a very astute question and something we get asked a lot, but we don't see it actually going into the brain, but we don't need it to.
Right. It's operating on peripheral things that then do get into the
Correct.
central nervous system. So yeah,
Exactly.
you're right. In that way, it's a very astute question. Um, so there are a number of other questions around eligibility. Again, I think going back to the clinic is going to be the most important thing. I think there are some questions like someone talking about a very slow progression beyond 28 months since symptom onset. Maybe you can clarify why for companies it's really difficult, especially in 24-week trials, to enroll people who are slower progressing individuals.
Yeah, no, very sympathetic to that situation. And so I kind of tried to allude to this, but from a perspective of like the goal of what, how we operate or when we design these trials, we have to be able to detect a difference, I guess. If we want to prove that this is effective and in the shortest time possible in 6 months, we have to make decisions. We have to have some idea of like how much patients are going to change so that we can detect a change. So unfortunately, in these clinical trials, we have to exclude very slow progressive patients simply because over a 6 month time period, it would be difficult for us to tell if there's a change in how that ALS is progressing. That's not to say we don't believe that it won't help patients in that, but for the purposes of the clinical trial, those patients need to be excluded so we can get the appropriate answer.
Great, thank you. I'm going to do one more, and then I want to dig into a little bit more about your clinical development plans, and a couple of other things. But one other question about side effects that we're seeing. I know I mean, these are well-tested drugs in different ways. I know there was a four-person study that was done by Dr. Appel and others. So maybe you could just allude to, at least the safety profile you have so far from the global knowledge of these
Yeah.
treatments.
No, great question. I'm very happy to report that it was incredibly well tolerated. So the major thing that people have had with this kind of treatment is that because anytime you do any kind of subcutaneous injection, you can get some irritation at those sites, which is why we recommend you rotate around them. But so far, we have not had any like serious injection
Adverse.
site reactions. There haven't been like serious infections. We're monitoring for those, but the low dose IL-2 has been tested in a larger populations in ALS in different studies and similar profile. The side effect profile, really like the only real thing that people have encountered or reported is some redness occasionally at the injection site, and otherwise feel pretty well.
Great, thank you. So I will come back to the Q&A. I know there are still a few things left in the Q&A. I did want to hit off on a couple of things. So I've been in ALS for quite a time now, and I've been fortunate to see Dr. Appel's work,
Yeah.
from initial Treg assessments, in terms of the promise that they had preclinically. There were some tests of drugs like fingolimod and, the Coya 101, where they're transplanting Tregs in. It was an extremely expensive endeavor that I know Coya tried to start moving into clinical development, and sort of improved upon the idea of it with Coya 302. And I know some of those trials that happened with fingolimod, with Coya 101 equivalent, also had very small open label clinical trials where there was a stabilization of people over 6 months. And when you look into the data, there's some interesting aspects to that. I mean, obviously there are a few people who seem to be progressing quickly and over the course of that. We also know that in ALS clinical trials that are open label and small, we often see a period where people can stabilize quite a bit. And it is so I just was wondering if you could first kind of clarify, because let me just say this. Years ago, Dr. Appel was on a video for ALS Association. He talked about stabilizing the disease, and it caused a huge uproar in the community, as it should, right? But I think we also have to just temper that. What this does, I think, is gives Koya good feeling that they want to move forward. It gives them good feeling to go and say, we want to invest in the next step in this. But also to, from your end, to maybe just temper when you show a graph that shows the stabilization, it's still very small, it's still open label, and it's still very exploratory.
Yes.
So you go okay.
Sorry, either of you.
Yeah.
Yeah.
Go ahead, Daniel, and then I'll go second.
Okay, sure. So I think that, the history of Koya, with 101, it was a cell therapy doing the exact same thing. Up Coya 101 was a cell therapy increasing the number and efficacy of regulatory T cells, literally taking those cells out of the body, putting them in a bioreactor, making them more efficacious and numerous, and putting them back. What we found, while there was significant signs that we could upregulate T cell number and function, so the biomarkers were showing efficacy, we found that there were certain patients, specifically those with C9orf72, the genetic variant I carry, that were not responding because the oxidative stress levels were so high in the body that when the reintroduced regulatory T cells were put back, so sorry, they were destroyed because of the highly oxidative environment in the body. So we're taking the same mechanism of action and then moving that forward with this combination therapy. That's, we said, okay, how do we do this in a more scalable way? So we found these off-the-shelf biologics and this combination that would overcome the scalability issue and the highly inflammatory issue. I think that also when we go back to these small open label studies, we need to look at the biomarkers and the change in biomarkers. Did we have the effect we wanted on regulatory T cell number and function? Did we reduce oxidative stress? Did we reduce these biomarkers that play a role in disease progression? ALS-FRS is something we want to hang our hat on, but in a small cohort of patients, it's hard to just focus on the ALS-FRS score. We need to look at the actual underlying mechanism of action and is it working? And because of the efficacy we saw in the fact that we understand that regulatory T cells are key to survival in ALS, and we saw that they were upgraded, we then decided that this was had legs to move forward to the phase 2.
Thank you.
Yeah, and to continue on that, and Daniel's been with the company longer than I have, and so he has some good insight into how that went. And you bring up a great point. So, based on the data we have, and always caveating that it's a small cohort, and taking that, for what it is, of course we hope for that. But we also at Coya are scientifically driven and we're data driven. So as we get the data, and we see what's going on, we can take a closer look. And like I said, we already mentioned one sub-analysis in terms of like what's happening with patients on standard of care therapy. When we get the data out from Coya 302, it is our full intent to look at the data objectively, see what we actually see, and then are there improvements, or is there a next step that needs to be considered? We fully believe that this combination is the combination that is going to have a great, a significant effect on the ALS. We hope for a halting. We also are prepared to at least slowing the disease progression. I think that there's enough data, proof of concept to suggest that that's the case. But as we, look at that, we are prepared for every scenario and are committed to try and find the right combination for patients in ALS to address this mechanism of action. So I can't speak to the future, but I can tell you that that we are looking objectively, and I think that we are with Dr. Appel's that there's legs to stand on there. But it's a great
Yeah.
question. So
Yeah, and I mean, and honestly, like I think there, coming from somebody who knows who's been involved in the ALS global landscape for a long time, you look at the MiroCals trial, which was just
yep.
IL-2, right? And also you definitely boosted the IL-2. Did we have better function of them? I don't
Hmm.
know. Was it in an environment where they couldn't function as well, which the abatacept will help with, hopefully?
Yep.
You look at histories like we had tegoprubart and we had other ones that were dampening the immune system, but you didn't
Correct.
have the extra bit that you get with the Tregs. So, I mean, moving it forward, it should be a strong assessment of whether neuroinflammation at all levels is
Yep.
something that can slow the disease. So I want to go back quickly to this again then. So in terms of um, next steps. I know that there's been some vocal about this potentially being a registration trial. It's also very small still. It's still arms of 40, right? And an ALS FRSR at 6 months is tough to be able to
Yeah.
detect that. I know you're looking for a big effect. Um, but
Yeah.
so let's say that you don't have a big effect. What will be the sort of go, no go parameters to think about maybe doing a larger registration trial? Or what would you then want to consider? Because I know, Daniel, you mentioned biomarkers. Like we won't have them, like neurofilament is one that maybe, if you have a significant
Yeah.
decrease in neurofilament, maybe there's something there. And then last, I'll say, because do you have any pre-specification to actually balance your arms by baseline neurofilament, since it's a little bit underpowered as well?
Yeah, no, so actually, and this is a feature we actually didn't talk about. We are probably actually one of the few trials that we are actually stratifying according to neurofilament. So as patients come in, we're ensuring that is to reduce the heterogeneity between each arm, and one of those is why, even though it's a small trial, we feel that it is going to reduce heterogeneity between the different arms and be able to give us like a full, a better assessment of the individual arms. We've done a lot of statistical modeling based on the expected rate of progression and the halting of the or the slowing of the rate of progression based on the previous data, taking into account the Miracal study and what we have from the investigator initiated trial. That we think that this is a great shot on goal. I can't tell you all of the decision benchmarks and thresholds, but we're having all of those discussions. We want, if this is, we believe that this is we have a strong belief that this is something that will greatly benefit the ALS community, patients with ALS, and we are going to take the opportunity to get it to patients and to people with ALS as quickly as possible. Whether that's sooner or later, we're committed to this and showing its efficacy. And I guess I can kind of leave it at that. But just so you know, we've spent a significant amount of time looking at those powering, balancing, using NFL as a stratification tool, and that we're committed to get this, if it's an effective therapy, to people with ALS as quickly as possible.
Great. Thank you. Yeah, and I know, and I love to hear that you're all about the science as well. And I know, at the end of the day, it's a partnership between, academia and industry and organizations in trying to figure out, what is something that, will ultimately get to the community. And of course, if there's anything that looks positive and
Yeah,
in a real way, we're all going to want to get that to people as widely as possible. Um,
right.
Okay, and so I'm going to ask one other question that came into the chat just because I want to make sure I hit as many of those off as possible. Was wondering just in the last minute if you could expound upon a little bit about why it's difficult to do expanded access programs, compassionate use programs. If you're a company that, maybe doesn't have a marketed drug, it's difficult, it's expensive, it is complicated, but that doesn't mean that you're not maybe a company that's interested in being community focused.
Yeah. So actually, I'll tell you that we are already investigating ways to get expanded access programs. So we're the lead PI for this study, academic PI, Dr. James Berry, a mountain in the field, and we're already in discussions with him about ways that we can get an expanded access program to participants with ALS. Of course, there are difficulties. We've alluded to this. If I had unlimited resources, of course, I would get that to everybody. But we're working diligently, and Daniel's on part of that, of designing and being able to provide the access to patients in an expanded way. Those that enter into the trial, we're committed to continuing to give the drug to them, and however we can. If they're participants in the trial, we are looking for ways to continue to provide that, as well as other expanded access opportunities. We're small but mighty, and we work day and night to try and get this out to everybody. So those plans are in the works.
And I'll say, we are grateful to Act for ALS and other funding mechanisms that have allowed for funding for EAPs. So it allows companies like ourselves, without a commercialized therapy, to run some of these EAPs. And hopefully that includes a larger patient cohort with different inclusion exclusion. So it's all in the works.
And just to note that much more difficult in Canada without the Act for ALS for those to happen. But
Correct.
those are the kind of conversations I'll just add to the audience. If you ever want to have those conversations, we do have a one-on-one at ALS Canada. We're happy to talk about expanded access stuff in Canada. But I just want to say we got to wrap it up here. Thank you so much, Daniel and Dr. Simkins, for being here today. We really appreciate the overview, the time to chat. Thank you, everyone out there who joined us today for ALS Clinical Trials Unboxed. We'll be back next month with another episode. And let's see where Coya 302 and the ALSTARS trial goes. We look forward to having it enroll quickly. We look forward to getting those results and seeing where the science leads us. So thank you so much, both of you. Thank you to everyone out there, and we'll see you at the next episode.
Thank you, everyone.
Thanks,
Bye.
David. Thank you all.
Thanks, David.